Welcome everybody to Tune In Tuesday number 10, our final Tune In Tuesday event of the year. So thank you so much for joining us. My name is Michaela Moss. I'm the Executive Director for the Friends of the Missouri Breaks Monument. We're a small nonprofit based out of Helena, Montana, dedicated to the protection and preservation of the Upper Missouri River Breaks National Monument through stewardship, education, and advocacy. 2021 has been a really big year for the Friends and for the monument. We both turned 20 years old this year. So we've been doing a bunch of different things to kind of celebrate this milestone. And one of them was to focus on education. And with COVID, we had to find a way to do that safely and connect people to the monument. So we've been hosting these online virtual education events throughout the year, kind of covering a whole swath of different topics related to the monument. And that was everything from the management, the ecology, the wildlife, how to recreate on the monument, how to hunt on the monument. And tonight we are really lucky to be continuing with more monument fun facts, and we'll be joined by Montana Fish, Wildlife, and Parks fisheries biologist Luke Holmquist. So we're really excited to hear more about Pallid Sturgeon from Luke, and thank you so much for joining us tonight. I will turn it over to him in just a moment, right after I go over some brief housekeeping about tonight's event. Everybody is, it looks like, muted right now. I will ask that you guys stay muted during the presentation just to cut back on the background noise, but there will be time for questions at the end of the event. So if you do have questions, then you can unmute yourself and ask anything that came up during the presentation, or feel free to type your questions into the chat whenever, and we'll get those answered at the end of the presentation. But other than that, I think I can end my portion of the chatting for now and turn things over to Luke to take it away.
Thanks, Michaela. So like Michaela said, my name is Luke Holmquist. I'm the fisheries biologist for Fish, Wildlife, and Parks on the Missouri River from Fort Peck Reservoir all the way up to Great Falls, and including some of the larger tributaries. So tonight I'm going to talk about pallid sturgeon ecology and recovery efforts in the Upper Missouri River. I've been working on and off in the monument for since about 2011. Did my master's work on pallid sturgeon, the monument, from 2014 to 2016. Briefly left to go do some pallid sturgeon work on the Yellowstone in Missouri downstream of Fort Peck, working with USGS. Then came back to my dream job here in Lewistown, where I get to continue working with this iconic Missouri River species. So just a little background of this talk is going to cover some background recovery efforts, ecology of the species, reasons for decline, as well as some of the research efforts that we've implemented, techniques used, lessons that we've learned, things we've learned about the fish, and then future work going forward, hopefully working towards recovery of this amazing species. So pallid sturgeon are a federally listed species. They're listed under the Endangered Species Act as endangered. They were listed in 1990. Throughout their range, they've experienced drastic declines. So the range of the pallid sturgeon goes from the Gulf of Mexico all the way up to the stretch that we're talking about today up here in the monument. It's a really wide range, but they've experienced, drastic population declines, mostly as a result of these dams that have been built on the main stem, Missouri-Mississippi River, and as well as some of the larger tributaries. So The recovery plan was established in 1993. They identified 5 limiting factors for the species: altered river connectivity and function, illegal harvest, water quality issues, entrainment, life history attributes. Up where we're talking about today, it's really a combination of these two: altered river connectivity and function as a result of those dams, and how those interact with the life history attributes of the Pallid Sturgeon. So the monument is found within what I call, and what the recovery team calls, Recovery Priority Management Area 1. So it's just Fort Peck Reservoir up to Maroney Dam. Maroney Dam is a dam that Northwestern Energy operates. They operate 5 of them in the Great Falls area. It is a barrier to movement, although naturally Pallid Sturgeon would have been blocked from going much further upstream by the falls. And then on the Marias River, Tiber Dam is located about 80 miles up from the confluence near Loma. It's a very beautiful stretch river, pretty natural looking, includes the Charlie Russell National Wildlife Refuge, obviously the monument, and a portion is also designated as wild and scenic. So a lot of protections are in place to keep this river as natural as possible. So in 1996, a couple years after listing, it was estimated that as few as 45 wild fish remained. And those 45 fish were about the size of the fish shown in this here photo. It's probably the only time that 4 of these fish were ever photographed at the same time in the upper Missouri River. And these 45 fish were all very large, and there were no small, recently produced offspring. So basically indicative that recruitment, when I refer to recruitment, I just mean baby sturgeon that are spawned and make it to 1 year old. No recruitment had been observed in the last 50 or 60 years. Which very closely aligns with when some of those dams were built on the Midstem Missouri. So we took those wild fish to the hatchery, like the good fisheries folks that we are, and initiate a conservation propagation program. And so we take those wild fish in, spawn them, and then release their progeny out into the wild. And so since 1997, we've released 16-year classes of these fish out there, 150,000 hatchery origin Pallid Sturgeon, or what I'll abbreviate as hops in this talk. And we actually were able to capture a lot of the genetic variability that was available in the wild population. So you'll notice that we used 50 females, 118 males to date. That's a lot more than the 45 I said that existed. A lot of those came from the population downstream of Fort Peck Reservoir in the Yellowstone, which are genetically the same population. They're just isolated from one another by Fort Peck Reservoir. Pretty monumental effort. Lots of hatcheries were involved, lots of people involved, but in the short term, this kept extinction at bay for at least the foreseeable future. Now, when I think of a conservation propagation program, in order for it to be successful, it has to fit certain criteria. So the first being the ability to collect and spawn wild fish, right? You have to be able to go get fish from the wild if you want to repopulate that stretch. And those fish that you stock have to be successfully adapted to the wild after you stock them. They must survive to sexual maturity in sufficient numbers, and then also have similar genetics and genetics as the wild fish that you're trying to replace, and also ultimately allow you to identify factors that are limiting recruitment. So like I said, we have successfully collected wild broodstock and spawned them in the hatchery. We captured a large amount of the genetics, the genetic variability that was available out there to us. We think 99% or so of the available alleles are captured in the hatchery progeny. Now, as far as do those hatchery fish adapt to the wild environment, there have been several studies looking at that. And we've also monitored those cohorts through time. We mark them all. We know who the parents of those fish were from the hatchery, so we can track them over time to see how those individual cohorts have done. And so we did a population estimate in 2016. There was about 4,100 individuals out there, give or take 600. They have really high annual survival after they reach a certain size. So you're probably thinking 4,000 isn't very many if you stocked 150,000. Well, about 110,000 of those were fingerlings. When they're really small, they don't survive as well. The bigger they get, they're kind of pokey, covered in scutes. Nothing really eats them after you get to a certain size. So, and then this figure shows it's just a length and age figure for those individual year classes of hatchery fish. As you can see through time, they are getting bigger. They're growing and surviving. So by all accounts, they are fairly acclimated to the wild environment. The next step being that they can survive to sexual maturity in sufficient numbers. So through the last 10 years or so, we've been collecting blood samples and gonad samples from these sturgeon, trying to evaluate when they become sexually mature. And that first year class of fish, the 1997 year class, which is kind of an iconic year class that I'll probably talk about a little bit. The earliest we observed was 14 years for males and 18 years for females. So they take longer than humans to reach sexual maturity. And we actually do have a number of those fish that are still immature at age 24. So they take a long time to reach sexual maturity. So it's a kind of a waiting game. You know, that's 24 years can be someone's career. Someone at the hatchery might never even got to see this come to fruition for even that first group of fish to reach sexual maturity. And then through that work, we also learned that, well, we kind of knew, but the hatchery fish, 1 to 2 years is the frequency at which they'll spawn. So males don't spawn every year and females don't spawn except for every 2 to 3 years. So a big female, upwards of 22% of her body mass can be eggs when she's running ripe. So it takes a couple of years to accumulate the amount of energy reserves needed to make those spawning migrations and accumulate that gonad mass. So survival of sexual maturity, they've started to become sexually mature. We have a little bit of lag time before the next year classes start reaching sexual maturity. So there isn't a lot of them out there, but there are at least enough that we can start to look into this last point on this list, that is identifying factors limiting recruitment, now that we have some reproductively active fish out there that we can track and see what they're doing. So in order to take a look at that, we first got to understand what a pallid sturgeon life cycle looks like. So if you recall, in 1996, the only thing that was out there was large mature adults. I like to point out to my parents that that's me in this photo, and I have matured as I got older. So those fish will spawn. They have a certain set of spawning cues that are required for them to spawn. They're really long-lived fish, and they have a mechanism kind of built into their life history, which is called follicular atresia. And so, like I said, 22% of their body weight can be eggs. A lot of energy there. If those spawning cues aren't there or conditions aren't right, these fish will actually reabsorb their eggs and get a jump start on the next cycle. So without those spawning cues, they won't spawn. But if the cues are there, they'll spawn. You get fertilized embryos. They're adhesive. So when the fish broadcast spawns over the substrate, they stick to rocks or a tree or whatever that fish is spawning near. And they stay there for somewhere from 5 to 8 days. And that's dependent on water temperature. And then they hatch. And when they hatch, it's basically just a yolk sac with a tail, and they're neutrally buoyant. They float downstream slightly slower than the current, again, depending on water temperature and water velocity. Can take anywhere from 9 to 17 days, 150 to 330 miles. And then at the end of that, they've absorbed all the nutrients in their yolk sac, and they have to settle out on the bottom of the river and start feeding on their own. And we call those exogenous feeding, young of the year, like this cute little guy on my index finger in this photo. And then if they make it to that point, hopefully they're able to grow, become juveniles. and then mature, and the cycle goes around and around. Like I said, that takes at least 14 years for males, 18 years for females. However, if you get your spawning cues are missing and you're stocking individuals, all you have is stocked juveniles and mature adults. So you need to figure out what those spawning cues are in order to get spawning to occur. So if we take just a look at what we've learned over the years, or at least some of this is maybe a little bit hypothesized, on what the spawning cues for pallid sturgeon are. The first one being photoperiod. So they spawn in the month of June or early July every year. Hard to mess up photoperiod. The amount of daylight stays the same on most dates year to year. Spring discharge, that one should perk some ears as far as the influence of dams. As you can see, this is just over on the right. That's just a series of hydrographs over the last 15 years. And so in the spring, when the snow on the prairie and the mountains melts, the rivers rise. When all that runoff is gone, they start to recede. So pallid sturgeon will migrate to their spawning sites on that ascending limb of the hydrograph. Male aggregations form at spawning sites. And then on the descending limb, as water's coming down, spawning, incubation, and drift occurs. And as the water's going down, it generally warms up. There's less water, so air temperature can heat it up a little faster. And then pallid sturgeon have a thermal optimum 18 to 22 degrees. They can spawn up to 24 Celsius, which is about 75 degrees Fahrenheit. Much above that, larval mortality starts to occur. Turbidity plays a role. So the Missouri River is a pretty muddy river. So that's a spawning cue as well. And then habitat, the appropriate substrate, velocity, and depth needs to be present. And then the presence of mates. One pallid sturgeon can't save the species. Needs to have another pallid sturgeon there. And then there's probably some other spawning cues, especially micro spawning cues that occur that we just don't know what's going on because we can't spy on a fish in a muddy river. So I alluded to the dams a second ago. So the impacts that these dams have on pallid sturgeon and a host of other fishes. There's a lot of other native species out there that depend on those natural hydrographs. So the main two dams that influence this stretch of river in pallid sturgeon is Canyon Ferry and Tiber's. Canyon Ferry is on the Missouri, located by Helena. And Tiber Dam is 80 miles up the Marias River near Chester, Montana. And they alter the hydrograph very expectedly by, in the spring, when that runoff comes out of the mountain and off the prairie, it gets trapped in the reservoir. So it lowers that peak flow that you would have had in the spring. And then they let that water out through the fall and winter. So you have higher base flows and lower peak flows. Also, dams can alter the temperature regime. Water getting released through the powerhouse generally comes from the bottom of the reservoir, where it's quite a bit colder than it would have been if it was flowing through a natural river. And so that altered temperature can last for a little ways downstream before it picks up a more natural thermal regime. And this is just figures showing how that looks for each of those dams, the top being Tiber. On the x-axis of these two figures is date, and discharge, or the amount of water coming down per second, the volume of water per second, on the y-axis. And the black line is what it looked like, the average before Tiber Dam went in, and the blue line being post Tiber Dam going in. So you see higher base flow and severely depressed peak flows. Now there's still a peak flow there, just not as dramatic as it was before. So maybe a little bit of a dampened spawning cue. And same thing on the Missouri River below Canyon Ferry. So that's the impacts of those dams upstream, but there's also an impact by the downstream reservoir. So Fort Peck Reservoir is the lower bound of the population here. And it's kind of the smoking gun for why we don't have natural recruitment for pallid sturgeon. So when the Missouri River flows into Fort Peck, it goes from moving stopped, essentially. It's still moving a little bit. But as water slows down, it doesn't have the ability to hold sediment or materials and move them downstream. So they all settle out in what we call the river reservoir transition zone. And all the plant material, dead fish, dead bugs, all that stuff settle out there, and the microbes, bacterial, all that good stuff actually consume it and use up all the oxygen. So when those drifting yolk sacs with a tail drift in there, there's no oxygen, they suffocate. So they all die when they go in there. So that's the impact of Fort Peck. Fort Peck is operated by the Army Corps of Engineers. Additionally, they can call for flood replacement storage at Canyon Ferry and Tiber. So in a year where there's flooding downstream, they can actually request that Canyon Ferry or Tiber holds back more water, potentially dampening the hydrograph on a year where you might have had a little bit more natural flow just because of the amount of water they were releasing at that time. So if you do get past spawning and you have larvae hatch and go into the drift, they end up in that anoxic zone and can't recruit and grow into a juvenile. So all we have is hatchery stock juveniles. That's kind of where we're at right now. So we've identified the factors that are limiting recruitment, but we really need to try and find a way to work around those factors and restore at least some recruitment. And the way we go about what I'm going to call recruitment monitoring is, first and foremost, we track spawning ready fish. So we go in the spring, we capture them, we do those reproductive assessments, and then we track them through the spring, identify spawning locations, habitat preferences, how far are they from that anoxic zone in Fort Peck. And then later we will monitor for progeny, kind of do this 3 different ways: larval sampling, which is basically anchor up with a boat and filter the current in these nets, trying to catch larvae as they're drifting away from those spawning locations. And then a little bit later in the fall, after spawning, we'll go out and perform what's called benthic trawling, basically dragging a big net on a frame across the bottom of the river, looking for those larvae or the young of the year. So fish that did successfully settle out before they reached the reservoir and died. We've never got one of those. And then also future monitoring. So every year we go out and trammel net and set line. So there's a chance that you pick one up from last year or the year before, or the year before that. And we do genetic samples on anything we catch during those and try to link it back to the genetic samples from the fish that we have radio tagged. So, and in doing so, we can evaluate differences in spawning location or spawning stats among years, compare discharge temperature, availability of mates, a host of other things to see what conditions might be suitable for getting some recruitment. The telemetry program has been ongoing since 2006. However, and then there was a little bit back in the 90s with some wild fish. However, it wasn't until recently that we had mature fish and we could actually look into some of these questions. The stuff in the 90s was just a couple of wild fish. Early 2000s, immature fish. We were answering some really good questions about what fish do post-stocking, but those fish just weren't mature yet, so we weren't able to do the spawning studies that we're doing now. So starting in like 2014, we really started to hone in on what pallid sturgeon do in the spring to spawn. So males and females do something a little bit different. This is just a simplification of how they migrate in the spring. So male pallid sturgeon might be spread out throughout the whole reach. Typically, there's a few more down lower in the system. As the river starts to come up, they start migrating, and then they typically end up in 1 or 2 locations down near Fred Robinson Bridge or the Woodhawk Campground, and they form these male aggregations, and they might be coming in and out. They don't just go sit in place, they'll move it. But there's these general reaches where the males kind of go and kind of think of it as they go and hang out at the bar waiting for the ladies to show up. So females also might start out spread out throughout the river, but instead of going straight to those spawning locations, they actually kind of go check them out a little bit. You know, they kind of do a drive-by, see what's going on, and they might go up and down past those spots multiple times before spawning, or sometimes, like I said earlier, they'll go atretic rather than spawning. But typically, spawning occurs down at those reaches, and actually we've confirmed hatchery origin fish having spawned 4 years in a row now, including 2019 when we actually did collect 4 free embryos, which is what the little yolk sacs with the tail are called. And we were able to link them back genetically to one of the females that we were tracking and thought had spawned. This was a pretty big finding for us because it was the first time that we definitely proved that the hatchery origin fish are spawning with one another, and they're capable of doing it. So it was really, really awesome. However, where those fish were produced, there's not enough drift distance. They drifted down into Fort Peck and died. So little Pallid Sturgeon larvae graveyard down there. It's pretty sad. But so we're constantly looking at ways that potentially we could get those fish to move upstream. And one of the things that has been suggested is a more natural hydrograph. And so this figure just shows the discharge, the river flows over the last 15 years in the Marias River, which is the blue line, and the Missouri River at Landusky. So you notice the Marias a lot of years doesn't even have a peak flow. It's just kind of flatlined a lot of those drought years. Last year was one of them. But the 2 years that really jump out are 2011 and 2018. So in 2011, record flows, a lot of flooding. However, we didn't have very many reproductively active fish out there to track. So there was a lot of water, but not really much on the telemetry side of it to make any conclusions. 2018, on the other hand, we did have a few. And if you'll notice, the Marias discharge in the Marias River was higher than we've seen in any of the other years of this telemetry study. And the Missouri River, second highest peak discharge and a pretty wide duration of flows being elevated above 20,000 CFS. So very, very interesting year. If we take a closer look at what was going on in the Marias in 2018, this figure X on our date on the x-axis, discharge on the left Y, and the solid line is the discharge. And then the dashed line is temperature, with temperature over here on the right. So in early April, we had a rain event at 10,000 CFS coming down the Marias is a lot. And then it came down and flows hung at about 3,000 CFS, peaked at close to 5,000. Then we're slowly coming back down. And then on June 21st, flows kind of coming back up a little bit. This dashed vertical line is when the Corps of Engineers called for replacement storage at Tiber. So releases were cut to 700 CFS. And temperature prior to that point had not reached in between these two horizontal lines, which represent Pallid Sturgeon spawning temperatures. So flows were cut before Pallid Sturgeon likely would have spawned. Why am I telling you this? Because Pallids did something interesting in 2018. So this figure is a little bit busy, but I'll walk you through it. Date on the x-axis. The right y-axis is discharge, and that's what those thick lines in the background are, with the Marias being the bottom one, and this one being the Missouri River. And then on the left axis, you have river mile. And so each of these colorful lines with dots on them is an individual female Pallid Sturgeon. And so when that peak flow in the Marias and Missouri happened, two of them took off like they were shot out of a cannon. And this line right here is Fred Robinson Bridge. Most of them were down around Fred Robinson Bridge in mid-April. And they went upstream past Judith Landing to the Marias River confluence. And then as soon as those lines go dash, those fish are actually in the Marias River. And so we had 3 females go in the Marias River, Another one came up late, was almost in there, hanging out around the mouth as flows were dropping from that 5,000 CFS. And then when flows were cut, everyone pretty much left and didn't go back in there. And they actually went down and spawned at that woodhawk aggregation that we were talking about earlier. And there were actually a couple of male pallid sturgeons in the Marias too at this time. So that was really, really exciting because if spawning occurs up here, you're 180 miles or more from that anoxic zone, and potentially you could get recruitment. However, because flows started to drop and water temperatures never reached the point where sturgeon would spawn, they left. So 2018 runoff, these two figures on the bottom are just the Missouri and Marias flows and how they compare to that pre and post dam hydrograph that I showed earlier. And what you'll notice is that similar magnitude to pre-dam flows, a little bit longer duration in the course in the on the Missouri side. The Marias, similar magnitude, truncated a little bit, the peak of flow maybe occurred a little bit early. However, we did have use of the Marias River during that runoff, but no spawning. So can really use that as a template going forward to, hey, can we get them back in there and maybe tweak a few things to get them to spawn. So going forward, the two big things that we're looking into on top of the monitoring efforts that we already have in place are these experimental dam releases out of Tiber and Canyon Ferry dams. And we actually did one in 2020. So basically we, in the spring, have communications with Bureau of Reclamation, looking at snowpack, forecasting how much water is going to be available. And then using that 2018 hydrograph, as well as the natural hydrograph before Tiber Dam, we build these test flows. So that's what this green line is what we recommended based on the amount of water that was there. And then the blue line is what we were able to actually get. There's a little more water than we were aware, and that got dumped out in July. Unfortunately, we only had a couple of mature females that were being tracked that year, and they spawned in the lower river rather than going up to the Marias. But we do have some other technology in place that I'll talk about in a second, where we tracked record numbers of shovel-nosed sturgeon and bluesuckers, another couple of species, long-lived native species that we're concerned about. So the flow test, although it didn't draw pallids in this time, pallids are kind of an umbrella species, which is just a term we use for a species that if you do something that benefits them, you're going to benefit a lot of other species. So really cool to be able to work with the Bureau and develop these flow tests, and hopefully going into the future, we can continue to do that and maybe find something that works to get pallid sturgeon into the Marias. And then we're going to do a larval drift test, which basically just means that we're going to go to the hatchery, spawn a bunch of sturgeon larvae and dump them into the Marias River where we think those fish would have spawned. And then we'll monitor to see if we have any recruitment from that event, which would validate that if spawning occurred, the Marias would be sufficient for recruitment, which might help us be able to push a little bit harder for a certain flow scenario. So those additional monitoring tools that we're also using that I just don't have time to get into today are pit tags, which is a similar, it's the exact same thing as when you chip your dog. So you put a chip in your dog and you can scan it and someone can return your dog to you because that code is unique to your dog. Well, we can do that with fish too. They're a lot cheaper than radio tags. So we can tag thousands of fish instead of, tens or hundreds with radio tags. They are limited to being able to use them in a tributary like the Marias or the Teton, just because of how the antenna has to be installed on the bottom of the river. But you can imagine, so this figure is just shovel-nosed sturgeon. The bars are the number of unique fish detected on a day at the mouth of the Marias River. and then flow overlaid. So you can see how as flows went up, a lot of fish went into the Marias River. So it works well for shovel nose. It's going to work well for pallid sturgeon. We've been tagging them for a number of years now. Really excited about that tool as a way to monitor use of the Marias. And then the larval sampling that I talked about earlier, actually saying, hey, this larvae was collected on this date and it came from a pallid sturgeon, pallid sturgeon, whichever pallid sturgeon we were tracking that day, being able to link that back just allows us to fine tune what we know about those spawning cues. And so the end goal, of course, being getting pallid sturgeon to spawn in the Marias River. They're not always all going to go there. They don't know that their progeny are floating into Fort Peck Reservoir. So some will always spawn at those Woodhawk and Fred Robinson Bridge aggregations. But hopefully someday we can get enough spawning to happen in the Marias River, or at least the upper Missouri, where drift distance is sufficient and those larvae settle out before they reach Fort Peck Reservoir. And therefore completing the life cycle without us having to stock hatchery juveniles. And with that, I'll take some questions. This is my black lab Hatch. He's my favorite larval sampling partner. He gets pretty into it. But yeah, I'll take any questions you have.
So Luke, it looks like we have a couple in the chat. So Bill asked, would it be correct that the Fort Peck anoxic zone also causes mortality in other species of fish and aquatic life?
It's possible. Not all fish have the same drift requirements. And we do see a fair amount of reproduction by the other native species. It's not a complete lack of recruitment. We do know that shovel-nosed sturgeon, the close relative pallid sturgeon, their larvae, if they spawn too close to the reservoir, their larvae don't make it either. Fortunately, shovel-nosed sturgeon are very good at utilizing the Teton and Marias River under a range of flow conditions to spawn. So a pretty healthy population of those two. So not to the extent that pallid sturgeon recruitment is affected.
Interesting. And William asked, are there any invasive species in this part of the river which may be impacting the survival or recruitment of sturgeon?
There are not any native or invasive species that we're aware of that are resulting in any impact to pallid sturgeon recruitment at this time.
Awesome. There are any other questions, feel free to unmute yourself and go ahead and ask or type them into the chat box. Now's your chance. We have another one. What does the paddlefish situation look like in that section of the Missouri?
The paddlefish population is actually doing pretty well. That's actually managed out of our Region 6 office. But they do a very close harvest. They closely monitor harvest in that stretch, and they have a stock assessment where they tag fish, monitor different year classes of fish and how they move through time, if they're showing up when they come up from Fort Peck Reservoir. And they really fine tune how many fish they allow to be harvested every year based on what their population estimates are. But they're doing quite well.
Good. Bill asked, doesn't recall whether you mentioned the average lifespan of sturgeon.
So the average lifespan is a tricky one because the only way to age a sturgeon is with its otoliths, its inner ear bones, kind of like counting rings on a tree. And so you'd have to kill it in order to age it. So I don't know specifically for my fish, but we've had similar size fish die in the past in the below Fort Peck population, and they've taken them in and aged them. But it gets kind of complicated because the older something gets, those rings get closer together and can be hard to tell. But they have done what's called bomb radiocarbon dating. So in the 1940s and 50s, when there's a lot of nuclear testing going on, there was atmospheric levels of carbon-14, I think. It's an isotope of carbon that's only really present when there's atomic bombs being tested. And anything that's alive is made up of carbon. So if you're alive during that time, you took on atmospheric carbon, that carbon-14 signature in your hard structures. And so they can actually see if that fish was alive in 1955 when the peak of nuclear testing was happening. And every single fish they did that with had that signature. So those fish were born before 1955 for the wild fish. And we still have some of those fish out there. So, 70, 80 years is not out of the question.
That's pretty impressive. And yeah, it's amazing what's below the surface that you don't know. Yeah. We had another question. Is there something special about woodhawk that draws them there?
So yeah, we think so anyway. So similar to like in the lower Yellowstone, that section seems to be kind of where it leaves the tight canyon and starts to open up a little bit more. And with that, there's a little bit more sand and gravel instead of the big cobble and boulders that you find upstream. So we think it's a substrate thing with more gravel and sand compared with that upper reach that's just boulders and big cobble.
We have another. Is FWP going to continue planting hatchery raised fish for the foreseeable future?
So we will, as long as the genetics of that fish are not already represented. We're not stocking fish just to stock fish. But if the genetics are not represented in this population, we will stock more fish. But we stock a lot fewer now than we did in the past, now that we have more fine-tuned survival estimates that inform us about how many fish we need to stock in order to get X number of fish to sexual maturity.
Gotcha. Nathan has a question about young sturgeons. I don't quite know what you're asking. If you want to unmute yourself. Maybe we'll get you in the chat. Okay, and then we had another. Are there any other reasons the shovel nose are doing better besides the spawning locations? Oh.
So they do have a shorter drift distance as well, almost like half the distance. We don't know why that is the case. So not only do they spawn in the Marias and Teton, but their larvae don't need as far to drift before they absorb their yolk sac. So it's kind of, they're willing to spawn further upstream and then they just don't need as long to drift.
Makes sense. Nathan, it looks like you.
Yeah, I'm just curious as to if there's enough water chemistry difference between the rivers that the young that are hatched up the Marias will tend to return up there. Like salmon do, they know their natal waters, they want to return to those. Is that happening here or don't you know?
Yeah, that's a great question. So we don't know. We don't know where the wild fish were born, and all of our hatchery fish were born in Bozeman, Isle City or Garrison. So hopefully they're not trying to get back there. We have released fish at a really young age at various locations. to see if maybe they go back to spawning or a stocking location. And they definitely don't go back to stocking location. So definitely that point where they imprint on their natal stream is kind of cut out at this point.
Okay, thanks.
We had another, do those oldest pallids continue to be fertile? Do you know?
For the most part, yes. We have had a couple of them where when they go to the hatchery really, really old, we aren't able to get as many good eggs out of them. But for the most part, yeah, they're still fertile.
Awesome. Dave and Arlene asked, why doesn't the Judith River play any role? Insufficient flow or size, maybe?
Yeah, that's an interesting question. The Judith has kind of always perplexed me a little bit in regards to why pallids don't go up it. My predecessor always joked that they only turn right, so they only went into the Marias. So actually this spring we did, our other biologist that manages the Judith, they did sample 2 young Pallid Sturgeon in the lower mile of the Judith. So they had some young fish that were using it. But we've never seen a radio tagged fish try to use the Judith at all. They frequent the area near the mouth. And I don't know if it's a substrate thing, could be a water chemistry thing, turbidity thing. We just don't know at this time, but it doesn't seem like they have as much of an affinity for it as they do the Marias.
Interesting, that's a good question. Get another comment. Bill said he invited Carrie Elliott from USGS to the presentation. And she sounded excited to hear what you were finding. She said she'd worked with you on the Yellowstone, and she did a presentation in 2020 called River of Sand. And there's a link in there if anybody wants to check it out.
Yeah, Carrie's awesome. I worked with her when I was with the USGS on the Yellowstone.
Thanks, Bill. Any other last questions? We have just a few minutes remaining if you have any others for Luke. Well, hearing none, we can start wrapping things up. Whoops. Sorry. Thank you so much, Luke. That was, I mean, I feel like I knew nothing about Pallid Sturgeon until now. So that was an awesome presentation. Thank you for so much detail and filling us in on so much of your research. I think I'm excited to hear more about it in the years to come when you guys find out a little bit more with these new spawning adults. So thank you again for joining us tonight. And thank you to all of you for tuning in. Of course, you are the reason that Luke and I are both here tonight. So thanks again for spending your evening with us. If you do want to rewatch this, there was a lot of information in there. So if you want to look back at some of those graphs or anything, we'll be posting this video on our YouTube channel in the next few days. And you can also visit all of our past Tune In Tuesdays on our YouTube channel. So if you get stuck inside on some of these hopefully cold, snowy days, we have plenty of stuff to watch. So hopefully all of these Tune In Tuesdays have provided all of you with a way to connect with the monument, even if you were not able to travel there this year or last. And we want to give a big thank you to Luke and all of our past Tune In Tuesday speakers for joining us throughout the year to share their expertise on all the different things that make the break so unique and interesting to all of us. So if you want to find out more about our organization, please visit our website at missouribreaks.org. You can find out more about our work, our partners, ways to join, ways to help and support, donate, all of that good stuff. Again, MissouriBreaks.org. And one more big thank you. The chat's filling up with excellent, great work, Luke. Thank you so much for your presentation. And again, thank you guys. Have a great night and happy holidays.
Yeah, thanks, Luke.